Dr. Moayed, the floor is yours to choose the primary amebic meningoencephalitis.
Assalamu alaikum. Thank you so much for having me again, the second year. So the topic I'm going to talk about is primary amebic meningoencephalitis, which is, I don't think anyone is an expert in this disease because it's so rare. But reviewing this topic was very interesting. So we'll talk a little bit about what is this disease and what is the epidemiology of it, the clinical presentation, how to diagnose patients, and how to treat them. And I'm going to start with a video from YouTube. The video is not scientific, but to be honest, the visuals are stunning, and I think it's worth watching, and I took the permission of the creator.
A war has been going on for billions of years that needs well-armed monsters who struggle with other monsters for survival. Having no particular interest in us, most of them are relatively harmless, as our immune systems deal with their weapons easily. But there are exceptions. Naegleria fowleri is an amoeba that has not only developed a deadly taste for human brains, but is also a match for our defenses and stars in dramatic headlines. What happens when this monster enters your body? Naegleria fowleri is an amoeba, a microbe with a nucleus, one of the smallest life forms on Earth. It is a voracious hunter of bacteria and other critters that it devours whole and rips into pieces. Like many amoebae, it is able to transform into different stages that help it survive. But most of the time, Naegleria fowleri is in its trophozoite stage, during which it looks like a squishy blob with tiny arms, and hunts, divides, and thrives. Its natural home is in fresh water, ponds, rivers, lakes, and hot springs. But, unfortunately, it also feels happy in pipes, swimming pools, fountains, or spas when they're not properly treated. The warmer the water, the more it thrives and multiplies. So in the summer, when humans seek to cool off and enjoy themselves, the chances are highest that both species will interact. Because this makes it hard to avoid, millions of people regularly have contact with the amoeba, especially in warmer climates, and many people even seem to have antibodies against it. And this is mostly okay. You can even swallow it without consequences. Things turn bad when people dive or swim in water contaminated with the amoeba and water splashes high up into their noses. In a single drop of lake water, there are millions of viruses, bacteria, and amoebae, and that isn't really a big deal. But Naegleria fowleri is different. Let's zoom into the nose of an unsuspecting victim enjoying a great summer day and see what happens. First of all, the amoeba doesn't really want to be inside your nose, as it's not really looking for trouble. It just wants to eat a few bacteria. Instead, it's greeted by your natural defenses. Unfortunately for humans, Naegleria fowleri happens to be exceptionally good at generally flying under the radar of your immune system. For example, the inside of your nose is covered by mucosa, a slime filled with chemicals that kill or stun possible invaders or alert immune cells. But Naegleria fowleri is not particularly bothered by them, and instead calmly checks out the scenery, mildly annoyed about the whole ordeal. Now, if you are unlucky, the tiny critter stumbles over something that actually sparks its interest. Nerve cells. Your nose is filled with a large network of olfactory nerve cells that pick up molecules from the outside and transmit their information to your olfactory bulb, the center of smell in your brain. To do their job, these cells talk to each other by releasing various messenger chemicals and recognizing them via specific receptors. One of the most important of these chemicals is acetylcholine. Through sheer evolutionary bad luck, Naegleria fowleri happens to have receptors that recognize acetylcholine, and it seems to attract them irresistibly, a little like moths that are attracted by light. So as your olfactory nerve cells do their job, using plenty of acetylcholine to talk to the brain, Naegleria fowleri enters your tissue. It seems to follow the chemical signals upstream. Neutrophils, crazy suicide warriors, begin to attack the amoebae. Individually, they have no chance against them, as the invaders are large and pretty buff fighters, used to dealing with tough enemies. So the defenders swarm the intruders and kill them either by vomiting chemicals that punch holes into them, or by literally ripping parts of them off and devouring them. But the Naegleria fowleri train is still on track, and while the neutrophil attacks slow them down, they continue to follow the olfactory nerves to their final destination, your brain. This process can take between 1 and 9 days, and you'll probably not notice anything during that time, until the amoebae reach the olfactory bulb, the centre of smell and entrance to your brain. Your brain cells are nothing more than helpless victims, and they all release that wonderful acetylcholine. Naegleria fowleri initiates a massacre and releases an onslaught of various attack molecules. Some of them are basically little bombs that rip holes into your cells on contact so their pieces can be easily consumed. But Naegleria fowleri is now multiplying, and it's also becoming really creepy. In a feeding frenzy, it can develop up to a dozen suckers called food cups that look like giant eerie mouths. The amoebae engage your brain cells, suck them in, and rip large bites out of them while they're still alive. Now things escalate quickly, and the disease that will kill you sets in. Alerted by the massacre, millions of immune cells, neutrophils, eosinophils, and microglias invade the infected tissue. Which is a problem. Your immune system is dangerous and not exactly a careful fighter. It's like burning down a forest to kill the wolves inside it. A really bad idea in the brain. They waste no time and attack the amoeba, using all the weapons available to them, from chemicals to trying to eat them alive. Neutrophils explode themselves to erect barriers spiked with deadly chemicals. A fierce battle ensues. Naegleria fowleri can actually fight back, itself attacking and killing many immune cells. The immune system now throws everything it has at the invader, but in vain. The complement system, tiny protein bombs that can kill intruders on their own, are easily disabled. Antibodies, usually one of your superweapons, are just destroyed or swallowed. A high fever that usually slows enemies down does nothing, as the amoeba actually thrives in the heat. All the while, the amoebae continue to multiply, fight, and devour your brain cells. A disastrous chain reaction is taking place. One major thing your immune cells do when they fight is to cause inflammation, which directs large amounts of fluid from your bloodstream into the site of an infection. So as the battle rages on without a clear winner, more and more fluid enters the brain. At this point, the human will feel symptoms that quickly escalate. It all begins pretty vaguely: a headache, fever, nausea, and vomiting. As the battle spreads rapidly through the brain, serious symptoms appear, from confusion, inability to concentrate, to fatigue, seizures, and hallucinations. The brain swells up massively, but can't expand due to the bones surrounding it. So it compresses and disables the brainstem that controls things like breathing. Usually within a week, the patient dies. Up to 97% of the patients infected by the amoeba share this fate. In almost all cases, by the time an infection by Naegleria fowleri is recognized, the disastrous battle for the brain is already so far along that there is almost nothing to be done. Not only do we currently not have effective treatments, there are also an abundance of open questions about how an amoeba that usually enjoys its life in open water is able to overcome our immune system so effectively. So how worried do you need to be about this horrifying killer amoeba? Well, not very. While the Naegleria fowleri is clearly extremely deadly, and the infection truly horrible, there have only been a few hundred cases in the last few decades. You are way more likely to drown in a pool than to get infected. Not only does the amoeba need to be flushed high up your nose, it also needs to get a good grip, and it also has to make its way through the first lines of your defenses. Ultimately, Naegleria fowleri is neither evil nor a huge public health risk. But every year, some unlucky people have to deal with it. We still have so much to learn about it, and until we find a way to treat it, Naegleria fowleri will continue to be this vague and horrifying thing, hunting in puddles and lakes, and sometimes pools. Usually for bacteria, and very occasionally for people.
Hopefully you enjoyed this video. Yeah, okay, so that's the fun part. Now we'll go to the scientific part. So, primary amebic meningoencephalitis is a rapidly progressive and often fatal condition caused by the free-living amoeba called Naegleria fowleri. It is a thermophilic bacteria in nature and commonly found in fresh waters. It was first identified in 1965 by Malcolm Fowler and Rodney Carter. So there is a different classification of this disease. So it's either confirmed when it is detected or identified in CSF or brain tissue using one of these methods, which is the immunohistochemistry or indirect immunofluorescence, PCR, or next generation sequencing. But if it is identified by other methods, including direct visualization, then it is called probable or suspected. And when there is symptoms, which is acute onset acute onset rapidly progressive meningoencephalitis within 14 days of exposure, then it is called probable, otherwise it is suspected. And all other cases by direct visualization is suspected, as we said. Distribution, there is around 381 cases detected so far as of 2022. 182 of them were confirmed. The rest is either suspected or probable. United States has 145 cases because of good detection, and 11 of them are case reports, while the rest, other than the case reports, are just environmental samples. The rest are non-US, but the detection rate is very low because of lack of ability to detect the organism. In Africa, it is underestimated and underreported, and this is uniform in all literature, that most likely there are a lot of cases there, but it's not detected. And Australia and the Indian subcontinent are hotspot for reported cases. The number of cases have been increasing since the identification of this organism by the rate of 1.6% per year. Also, the number of cases detected ante-mortem is even increasing compared to post-mortem because of improved detection. The exposure is associated mainly with the swimming and diving in fresh water, and the most common body of water is lakes, pools and reservoirs, not the water activities, I mean. And this is the same when it comes to US versus non-US, while the cases that has been detected from sources other than natural lakes and ponds is higher in the non-US. And this is most likely related to the regulations of water control and sanitation. When you see also the gender differences and the demographics, most of the cases are among male, and the majority of patients died, and this is why women live longer than men because of the water activity. So in clinical presentation, usually patients, the incubation period is around 6 days, and the median duration of hospital stay is around 3 days for those who died, and around 30 days for survivors. And the time to death is usually within 5 days. So symptoms the disease has two phases. The first is the flu-like prodrome, which is associated with fever and headache in almost all cases, while late presentation are usually associated with altered mental status and a picture of meningitis. In the testing, the CSF analysis usually has high opening pressure, high WBC, high RBCs, high WBC that is mainly neutrophilic, and high protein with low glucose, but it is mainly neutrophilic, not lymphocytic. The CSF analysis also changes in color as the disease progresses, from gray to yellow-white, then red, and this is due to subarachnoid hemorrhage, which is associated with the progression of the disease. And in conjunction with the CSF, usually patients in their CBC would have peripheral leukocytosis with the predominantly neutrophilic shift and band cells. The wet mount of the CSF done immediately after the CSF analysis is of paramount importance because the parasite can stick to the tube. So if it is saved in any environment, it has to be processed before checking it. So it has to be agitated, and also sometimes centrifugation may concentrate the amoeba at the bottom of the tube before it is tested. And the stain of choice is not gram stain, which is a routine in CSF. But if you suspect it, you should ask the lab to do GIMSA or right stain. And this method, although it can detect the organism, does not differentiate the Naegleria fowleri from other similar diseases with similar presentations, which is Acanthamoeba species and Meningoencephalitis. So the other modalities include PCRs, which is the gold standard. It is very specific and also can differentiate the different species of Naegleria, because only Naegleria fowleri is pathogenic, while there are maybe around 6 other species that are non-pathogenic. And the other molecular testing called loop-mediated isothermal amplification assay, which has been tested in a few cases with good results. Antigen detection is very sensitive, but it doesn't differentiate species of Naegleria, so it can detect contamination sometimes. And ELISA is available, but never been tested in any case. Serology using immunofluorescence-based antibodies in serum is also a good test, and it is sensitive, but unfortunately it may persist for up to 4 years in some of the survivors, and it's not suitable for diagnosis because the disease progression is so rapid. Usually patient does not mount enough antibody response for this test to be used, and it does not distinguish between acute infection and previous exposure, and it is time consuming. Finally, the advanced molecular techniques, which include whole genome sequencing or transcriptomic and proteomic analysis like multitome. So regarding treatment, all of the data regarding treatment is extrapolated from case reports and from personal experiences of experts, and there are no studies done on this disease because of the rarity. And the CDC actually recommend the following regimen, which is amphotericin B at 1.5 milligram per kg per day, divided in 2 doses, intravenously for 3 days, followed by 11 days of 1 milligram per kg per day, once daily. And another regimen of amphotericin B intrathecally for 2 days, followed by 8 days at a different dose. This is to be combined with azithromycin, dexamethasone, fluconazole, miltefosine, and rifampicin. And the intrathecal amphotericin B is only kept for patients with evidence of high intracranial pressure. So these are important definitions we're going to use regarding when we talk about how to choose the treatment options, because we don't have enough data, so they used to study this disease, looking at the frequency of used medication and the efficacy of used medication in survivors. And then I'd like to point at these two abbreviations, GAE and BAE, which is granulomatous amebic encephalitis, which is caused by Acanthamoeba, and Meningoencephalitis amebic encephalitis, which is caused by Meningoencephalitis, because we're going to talk about them a little bit next. So all medications have low efficacy, okay, for all medications. And the best one that showed efficacy when used was rifampicin at 24%. But rifampicin had efficacy of around 50 to 60% when it was used in granulomatous and Meningoencephalitis-associated amebic encephalitis. So the disease progression is very rapid in primary amebic encephalitis, while it is a little bit slower in the other diagnoses. Amphotericin B is the most frequently used medication at 70% of the cases, and only 19% of those patients survive. The conventional amphotericin B has lower MIC to Naegleria fowleri, and it is preferred over liposomal, although liposomal does cross the blood-brain barrier better. Intrathecal amphotericin B is suggested only for cases with increased intracranial pressure. Okay, 2 slides. So rifampicin is the second most frequently used medication at 48%, and has no significant activity in vitro, but it has the best efficacy in clinical cases. The efficacy also thought to be synergistic with amphotericin B. Fluconazole is the most common used azole at frequency of 36 and efficacy of 17%, and has an in vitro activity along with itraconazole against primary amebic meningoencephalitis. Azithromycin does cross the blood-brain barrier and exhibits synergistic activity with amphob in vitro, with frequency and efficacy of around 20%. Miltefosine was initially developed as an anti-cancer chemotherapy, but repurposed for treatment of leishmania, and this is why they used it in these cases. And it did show in vitro activity against all free living amoebae, but the only 3 survivors received miltefosine in primary meningoencephalitis, but it is not in use anymore. Nitroxuline, which is a quinolone derivative that showed in vitro activity against Naegleria fowleri, it was not tested yet in cases, but it was successfully used in Balamuthia-associated Amebic encephalitis. Finally, maintaining adequate cerebral perfusion and reduction of intracranial pressure is paramount to success of therapy, and mild to moderate hypothermia, 32 to 34 degrees, may be beneficial to improve survival. And in summary, the disease is very rare, and the risk of acquiring the disease is actually very low, so it doesn't cause risk, but without high clinical suspicion, you will never detect such a disease. It is preventable by avoiding diving and water activities in stagnant water, and if it has to be, at least no nasal plugs has to be used. Once suspected, confirmation should be sought before treatment because mortality is very high. And treatment is available, but we should follow the guidelines and seek help of the experts. And thank you so much.